So we will now start with the presentation of the last group, which is group 6. Group 6 has Ishan, Shamika, Samiksha, Rishali and Maitreya. So you guys can cast the screen and begin with the presentation.
Sir, can I start?
Yes, yes.
Yes. Thank you, sir. Good morning, everyone. This is Maitreya Naik. I'm a part along with Shamika, Samiksha, Trishali, and Ishan. We are going to speak about the central nervous system. Let us first have a look about the details of the CNS. The central nervous system consists of the brain and the spinal cord. It is referred to as central because it combines the information from the entire body and coordinates the activity across the whole organism. The brain is protected by the skull, the cranial cavity, and the spinal cord travels from the back of the brain to the center of the spine, stopping in the lumbar region of the lower back. The brain and the spinal cord are both housed within a protective triple-layered membrane called the meninges. Fun fact about the CNS: the brain consists of estimated around 100 billion neurons, with each connected to thousands more, and uses 20% of the total oxygen that we breathe in. The CNS can be roughly divided into white and gray matter. As a very general rule, the brain consists of an outer cortex of the and in the area housing parts of the white. Both types of tissues contain of glial cells which protect and support neurons. White matter consists of axons and oligodendrocytes, a type of glial cell, whereas a gray matter consists predominantly of neurons. Now, let's get into details of the system. First, I'll talk about the brain, followed by the spinal cord. The brain is the central control module of the body and coordinates activity from physical motion to the secretion of hormones, the creation of memories, and the sensation of emotion. However, many higher functions like reasoning, problem solving, creativity involve different areas working together in the networks. The brain is roughly split into four lobes. First, the temporal lobe, important for processing sensory input. Occipital lobe, visual processing region of the brain. Parietal lobe, the parietal lobe integrates sensory information, for example, touch. Frontal lobe contains the majority of the dopamine sensitive neurons and is involved in attention, reward, short term memory, etc. Now, we will have a look at some specific brain regions in little more detail. First, basal ganglia, involved in the control of voluntary motor movements and the procedural learning and decisions about which carries out the motor activities. Cerebellum, mostly involved in the precise motor control but also in language and attention. Broca's area. This small area on the left side of the brain is important in language processing. Corpus callosum, a broad band of nerve fibers that join the left and the right hemispheres. It is the largest white matter structure in the brain and allows the two hemispheres to communicate. Medulla oblongata, extending below the skull, it is involved in the involuntary functions such as vomiting, breathing, etc. Hypothalamus, sitting just above the brainstem and roughly the size of an almond, the hypothalamus secretes a number of neurohormones and influences body temperature control, thirst, and hunger. Thalamus. The thalamus receives a sensory and motor input and relays it to the rest of the cerebral cortex. It is involved in the regulation of consciousness, sleep, etc. The cranial nerves are 12 pairs of the nerves that arise directly from the brain and pass through holes in the skull rather than traveling along the spinal cord. These nerves collect and send information between the brain and the parts of the body, mostly the head and the neck region. Of these 12 pairs, the olfactory and the optic nerves arise from the forebrain and are considered part of the central nervous system. The olfactory nerves transmit information about odors from the upper section of the nasal cavity to the olfactory bulbs on the base of the brain. The optic nerves carry visual information from the retina of the primary visual nuclei of the brain. Each optic nerve consists of around 1.7 million nerve fibers. This was all about the different parts and specific function of the brain. Now we move on to the spinal cord. The spinal cord, running almost the full length of the back, carries information between the brain and the body. From the brain stem, where the spinal cord meets the brain, 31 spinal nerves enter the cord. The length of the spinal cord varies from person to person. According to some estimates, females have spinal cord of about 43 centimeters, while males have a spinal cord about 45 centimeters. Along its length, it connects with the nerves of the peripheral nervous system that run in from the skin, muscles, and joints. The spinal cord contains circuits that control certain reflexive responses, such as involuntary movement of your arm might make if your finger was to touch a frame. The circuits within the spine can also generate more complex movements such as walking. The spinal cord comprises three parts: the cervical, thoracic, and the lumbar regions. These were the various details of the central nervous system. Now, Shamika is going to explain about the infections associated with the system.
Hello. So, I'll be talking about infections of the central nervous system. As one of the most important defense mechanisms of CNS is blood-brain barrier. The blood-brain barrier functions to maintain hemostasis in the brain through restricting the flow of chemical constituents from the blood to the CNS. In order for the CNS to become infected, the bacteria, parasite, or the virus, the blood-brain barrier must be penetrated. The infections caused by the blood-brain barrier are, by the organisms are meningitis, encephalitis, and brain abscesses. Meningitis is a inflammation of the three membranes which together are called meninges. Meningitis is caused by range of different bacteria, fungi, and viruses. Once the blood-brain barrier is disrupted, these organs gain access and multiply within the CSF, a site which is a site initially free of antimicrobial antibodies or phagocytic cells. As the barrier keeps the large molecules, toxic, and most blood cells away from the brain, when disrupted, there will be an increase in the amount of white blood cells, red blood cells, immune system chemicals, toxins, proteins, and other microorganisms which cause inflammation. The other routes of infections are skull injury, medical surgeries, etc. Uh, Bacterial infection, which is also called as septic infections, the organisms that cause bacterial meningitis may live in the nose and the throat. Bacterial meningitis can spread through contact with saliva and nasal droplets. The infection without any pronounced neutrophilic response, fungal viral infections, are called aseptic meningitis, also called as viral meningitis. The types of meningitis is is relatively common. This type of meningitis is relatively common and it can be serious. It is caused by a variety of different viruses. Viral meningitis is usually mild and antibiotics are not needed. But bacterial meningitis is a medical emergency and can be fatal if not treated quickly. The neonates and the patients with prosthetic devices, particularly CNS and ventriculoperitoneal shunts, are at increased risk for getting meningitis. Next is encephalitis. Encephalitis is an acute infection of the brain. Uh, Most cases of encephalitis are caused by viruses. Access to the brain by the brain is by blood or nerves. Once the virus have gained access to the bloodstream, they can multiply and spread through the to the brain and the spinal cord. After breaching the blood-brain barrier, the viruses slip inside the blood cells. This disrupts damage and ultimately ruptures the infected brain cells. Certain viruses have a preference for different areas of the brain. For example, the herpes simplex virus likes to target the temporal lobes located near each ear. The body notices the invasion and mounts an immune system response. This causes the brain to swell. The combination of infection and immune response creates the typical symptoms in viral encephalitis. Uh, these are the causative agents of encephalitis. As arbovirus, it spreads through infected mosquitoes as through the bites direct into the bloodstream. Encephalitis with meningitis is known as meningoencephalitis. This is a rare rare and this is caused by uh, this cause This is caused by Nelaria foliae and Acanth amoeba species. These organisms are acquired during swimming or diving in natural lakes or ponds. The prognosis for a person with encephalitis varies according to the age, like a very young age or very old. These people are at high risk. Uh, and and the particular virus that causes the disease. While people may recover, in severe cases, the illness may be fatal or result in brain damage. Next is brain abscesses. A brain abscess is a pus filled swelling in the brain. It usually occurs when the bacteria or fungi enters the brain tissue after an infection. There are three main ways of brain abscess development. First, an infection in other part of the skull, like ear infection or dental abscess, which spread directly to the brain. Second, an infection in another part of the body, for example, the infection that causes pneumonia and spreads into the brain via the blood. Third, trauma such as a severe head injury, the cracks open the skull, allowing the bacteria and fungus to enter the brain. Brain abscess is regarded as medical injury. Swelling caused by abscess can disrupt the blood and oxygen supply to the brain. This also There is also a risk of abscess disrupting. Uh, if if brain abscess is left untreated, it can cause permanent brain damage and could be fatal. Now, Samiksha will talk about sample collection.
Am I audible? Hello.
Samiksha, you have to speak louder. We can't hear you clearly.
Now I am audible. Hello.
Yes.
Rishali, could you focus on the lumbar puncture image?
Yeah,
Hello.
I did. My internet is lagging a little bit. I'm sorry.
Okay. So as we all know that CSF, the cerebrospinal fluid, is the most common sample collected to find out the causative agent which is causing any CNS related infections. Uh, CSF is collected aseptically by a medical. I can't see the image. Hello.
Yes, you are audible, but please be a little bit louder. Yes, you are audible.
Yes. So CSF is collected by a medical procedure called as lumbar puncture, uh, it, which involves uh, the collection of CSF. In lumbar puncture, the patient is kept motionless and is the patient is kept motionless in a position such as sitting position or lying down on a side, and the patient is told to arc forward so that the lumbar vertebrae are visible, after which skin is disinfected using alcohol, and the spine needle is positioned between the L4 and L5 vertebra, and then the needle is inserted into the skin and the CSF is collected from the subarachnoid space, after which the insertion site is sealed. Approximately 5 to 10 ml of CSF should be collected into three sterile tubes, in which the first tube goes for glucose and protein, the second tube goes for the culture, and the third tube goes for the cell count. For the transport of CSF ideally should be delivered to the laboratory immediately or within 1 hour. If that's not possible, we can use a trans isolate medium, which is also called as the TI medium. It is used for inoculation medium and as well as for transport of CSF. Uh, Trans isolate medium is basically a biphasic medium consisting of a solid phase, which is a charcoal starch agar, and a liquid phase, which is tryptic soy broth. This medium supports the growth of organisms like N. meningitidis, S. pneumoniae, and H. influenzae. The next sample which we can collect is the biopsy specimen. Mostly it is a pus sample or aspirate, which is collected in the case of brain abscesses. The procedure involves this biopsy surgery is done in a hospital operating room. Before the biopsy is done, CT scan or MRI of the patient in 2 or 3 planes is taken to locate the abscess. After which, the neurosurgeon makes a small incision, few millimeters long. Then the needle is insert. Then the hole is drilled into the skull. After which a needle is inserted and the sample is taken out. After which the area is sealed. Uh, the samples, the abscesses or the pus sample should be transported to the lab immediately and in strictly in an anaerobic conditions. So we can transport the sample in anaerobic transport medium, which is also called as ATM, or we can use sterile saline to transport the sample. ATM is a medium which has contents like sodium thioglycolate and buffered mineral salts which provide the reduced environment. Is It is also having a resazurin indicator which turns pink in presence of oxygen. If there is any oxygen, the resazurin indicator will turn pink. Next, if we don't want to use the anaerobic transport medium, we can also use there are commercially available anaerobic transport devices which we can use. So the device, Rishali, can you zoom in? Hello, hello, Rishali.
I did, Samiksha.
Oh, sorry. Uh, so the anaerobic transport device is basically like a test tube. Uh, we have to collect the sample in the tube C, which is labeled which is called as the central tube. After the collection of sample, we immediately have to seal it with the plunger that is denoted at denoted as A there. So after that sample is collected, we have to push the sample into the rubber stopper, and then when we push the plunger, the tube is pushed down into the outer tube of the device. So the environment in the device is made up of gases containing CO2, N2, and H2 gas. If there is any oxygen in the sample, it will be removed as water in the presence of palladium catalyst. And there is also resurgin indicator in this device, so that uh, we can see if there is any oxygen, it will turn pink in the presence of that. Next sample we can collect is blood samples performed by venipuncture method to compare with the CSF samples, and to compare the like the procalcitonin and the uh, white blood cells count. If there is any infection, we have to compare the both. And in some cases we can also collect the nasopharyngeal swabs, like in case of bacterial meningitis or encephalitis. Thank you. Rishali, you can continue.
Am I audible?
Yes.
Okay. So I will be speaking about the precautions and gross sample examination. So let's begin with the precautions. This procedure must be conducted in, let's start with the cerebrospinal fluid. The extraction of cerebrospinal fluid must be conducted in strictly aseptic conditions to avoid the contamination of the CSF. To ensure this, the skin of the patient is first cleaned by an antiseptic. Disposable materials are preferred over reusable materials. However, hemocytometer and pipettes reusable hemocytometer and pipettes are used in hospitals frequently. So, contaminated reusable materials will be soaked in 70% alcohol for disinfection. Disposable items and spinal fluids will be considered as biohazard and should be disposed of as such. So, Samiksha talked about how 3 tubes of CSF are taken. Here, it is to be noted that the first tube can never be used for microbiological culturing because the first tube will contain a lot of contamination from the skin of the organism. Okay, sorry. This sample should not be refrigerated. It should be maintained at RT and should be processed as quickly as possible to ensure to avoid the rupturing of any blood cells that are present in the CSF. For biopsy, standard protocols are followed and aseptic conditions are maintained. The sample must be fixed with 10% formalin. The volume of the formalin should be at least 20 times as much as more than the specimen. If there are several lesions or tumors present, the sample should be taken from multiple places to ensure proper sampling. Coming to the gross specimen examination, the approximate quantity of the sample of each tube should be noted. CSF is typically colorless. The presence of red color may have been caused by a traumatic tap during the sample collection, which is when a blood vessel was harmed or ruptured during the extraction of the sample, or by a recent hemorrhage in the patient. In case of yellow CSF, this condition is known as xanthochromia, and it is caused by bilirubin present in the CSF. This can be caused by an infection, a recent hemorrhage, or severe systemic jaundice. The CSF is typically clear, and any turbidity can indicate an increase in the number of WBCs or abnormally high protein levels. This indicates an infection. The presence of clots. In case webbed clots are present in the CSF, uh, a tuberculosis infection may be present. A clot may also be seen in the case of a traumatic tap, but since the clot will not be webbed, it can be easily differentiated from the tuberculosis infection. In case of a biopsy, the size of the specimen should be noted in form of its largest dimension. It does not play a major role in the diagnosis of the disease, but it does give information to the neurologist about how well the sample represents the lesions and tumors present in the patient. The site from which the sample has been collected plays a major role in diagnosis, so it should be noted. And specimen handling, while specimen handling is relatively fixed, it should still be noted because in many cases, there is a deviation from the standard protocols. So instead of 10% formalin, for example, sometimes 70% alcohol may also be used. It will be valuable information to the physician, which is why it should be provided. Now, Ishan will continue with the sample processing.
Uh, Thank you, Rishali. So, before going on to sample processing, let us first understand why there is so much emphasis given on proper handling of specimen and proper processing. Why is it so important? The reason being, first, is to eliminate or reduce the exposure to humans and the environment. Second is to ensure accurate diagnosis and thereby save lives. And third is to maintain the integrity of a specimen or sample. In case of meningitis or encephalitis, as previously mentioned, CSF is used as a sample source. Once it is collected and transported to the laboratory, there it is centrifuged at about 1000 G for about 10 to 15 minutes, beyond which the supernatant is used for rapid diagnostic tests, chemical analysis, and serological analysis, while the sediment or the pellets are used for staining purposes and for isolation on culture media. This is the case if the sample has been transported to the laboratory within the stipulated time of 1 hour. However, if the sample is transported beyond 1 hour, then the CSF is inoculated in trans-isolate medium and incubated at about 35 to 37 degrees Celsius. The CSF is then used as an aliquot to carry out the further process. Coming to brain abscess or biopsies. Here, rather than using the CSF, we use the abscess or aspirate as the sample source for examination. The reason being, in case of CSF examination, the findings are too vague. Here, the biopsy samples are homogenized before plating and smear preparation. Importantly, this processing is kept to the minimum to reduce any sort of oxygenation and thereby maintain anaerobic conditions. Like in the case of meningitis and encephalitis, here the samples are plated on blood and chocolate agar plates, incubated in about 5% CO2 for about 3 days. However, in addition to this, an anaerobic agar plate and cloth with an indicator and certain nutrient sources are also inoculated and incubated to ensure that we maintain an aerobic environment. Vishali, could you move up to the other? Yes. So now let us understand the different laboratory tests or diagnosis that are carried out in case of CNS infections with the primary focus on meningitis. As mentioned previously, in gross examination, we study the color, clarity, presence of blood, clots, etc. in the CSF. The normal range in cell count studies for WBCs is 0 to 5 cells per mmq, while for RBCs it is 0 cells. In case of meningitis, this number shoots up to about 100 cells per mmq, and in some extreme cases, it can even increase to 1000 cells per mmq for WBCs. Similarly, for differential count, the normal range of lymphocyte, monocyte, and neutrophils are 60, 30, and 2% respectively. This is especially of crucial importance when talking about bacterial or viral meningitis. In bacterial meningitis, there is an increase in the number of neutrophils, while in case of viral meningitis, the lymphocyte count is elevated. In chemical analysis, we have protein analysis and glucose analysis. The normal range for protein analysis is 15 to 45 mg per deciliter. The four most common proteins that are present in CSF include prealbumin, albumin, transferrin, and IgG. The CSF glucose content is about 60% in comparison to the blood glucose level. Now, the microscopic examination involves gram staining or acid-fast staining if we suspect an infection by Mycobacterium tuberculosis, or even India ink staining when the meningitis is caused by Cryptococcus neoformans. In case fluorescent microscopy is available, we can also use acridine orange for staining. This helps when the bacterial number is extremely low and thereby increases the sensitivity of the test. As far as culturing is concerned, we use blood agar and chocolate agar with 5 to 10% of CO2 for adults and McConkey's agar for neonates. The first two are kept under anaerobic conditions, while the latter is placed under aerobic conditions. In accordance with the microscopic examination, if we suspect an infection by Mycobacterium tuberculosis, Algae medium is used, while if there is an incidence of fungal meningitis, Sabrot's dextrose agar is used. Serological testing involves latex agglutination test, which involves rapid bacterial antigen detection. PCR is a highly specific and sensitive method to detect the pathogens in the CSF sample. This can be the sample can be taken directly from the CSF or from the CSF which we have inoculated in trans isolate medium. Lastly, bacterial meningitis is associated with bacteremia, and hence blood can also be used as a culture source. And here these are pictures beginning from the left is Neisseria meningitis on blood agar plate. In the next picture there is the staining of Neisseria meningitis, where we can observe the diplococcal arrangement of Neisseria and surrounded by human cells, basically the leukocytes. Below that, we have the India ink staining of Cryptococcus. These cells, oval round-shaped cells, are typical of Cryptococcus. It is an encapsulated organism, so it has this large polysaccharide containing a capsule, which can be seen, which in this case as colorless in case of the staining. And lastly, we have a brain biopsy specimen for cerebral abscess. So this is all from our side, and these are the references used by us. Thank you very much.
Okay. Good job. Can you go to the central nervous system anatomy, please? Others can start typing their questions in the chat box. Okay, so Maitreya, can you tell me what is the role of corpus callosum? Hello, Maitreya?
Yes, sir. Yes, sir. Yes, sir. Sir, it basically it helps in joining the left and the right hemispheres of the brain, and it has the largest part of the white matter. Like I mentioned, like the central nervous system has the white matter as well as the gray matter. It has the largest part of the white matter structure in the and basically the children having problem in corpus callosum faces are like basically dyslexic, and they have a smaller corpus callosums. Uh, yeah, that's all I know about that.
Okay, okay. Uh, can you go to the section on infections of CNS? Okay. So, other than bacteria and viruses, are fungus and protozoa also involved in causing meningitis? Whoever dealt with this section? I think Shamika spoke about it, right?
Hello, sir.
Yes.
Yes, sir. Other than bacteria and viruses, it is also called by fungus and parasites. In fungus, it is caused by Streptococcus neoformans, and parasites, it is caused by a free living amoeba, Negleri foliae.
Okay. Okay. And can you go to the part of just above the sample collection and transport? Okay, it's about encephalitis. Okay, okay, ठीक है. Sample collection पे आ जाओ नीचे. So, Samiksha, when you talk about sample collection and you speak about biopsy, can you tell me which are the possible infections in which biopsy is done? For any infections, do you think biopsy is performed?
Hello?
I'm audible, right?
Hello, sir. Can you
Ah,
repeat?
yes.
Hello?
Bolo, Samiksha. We can hear
Yes, you
you.
are audible.
Sir,
Just
can you
continue.
repeat the question?
I asked that which are the infections in which you perform biopsy?
Oh, so in case of brain abscess infection, which is mostly caused by anaerobes.
Okay,
Hello.
for that you perform biopsy.
No, no. For that we collect the pus sample. We do this procedure.
Okay, okay, okay. Fine. Uh, can you go to the next part? Sample processing part. So Ishan, many a times, it may happen that the CSF sample that has come to the lab, that might be inadequate. So in those circumstances, have you come across any methodology to deal with or to work with the bare minimum amount of CSF sample that has been attained?
Yes, sir. For that is for the for that is the basic reason why we are using 4 tubes to collect the sample. Actually, as Samiksha mentioned, apart from we also have a fourth tube which is used as a sort of a special specimen control in case something goes wrong. And further, it actually depends on the uh, uh, person performing the test and the physician to take a call as to uh, what needs to be done next, or in case the sample can be like outright rejected as well.
Okay, okay. So there are questions in the chat box. You can start taking them up.
So, Saloni, regarding the exhaustion of the sample, you have to ensure that you take, as Ishan mentioned, you take a fourth tube as well in case of large sample analysis. So, if you suspect something, a parasitic or a fungal or a mycobacterium infection, you will take a fourth tube as well on which you can conduct extra tests. So, this is to ensure that you don't run out of the sample. And Nitin sir, the standard volume recommended is 3 to 4 ml per tube. And sir, what do you mean by the significance of the Nitin sir? Oh, okay.
Yes, we just discussed about it. That running out of the sample and okay, and obviously, if you go for the higher amount of withdrawal of that CSF, that can also have some of the consequences as far as the health of that patient is concerned. Okay, that's what I expected. Yes. Yes, others can go for the answers for remaining questions in the chat box.
I think there are still questions left to be answered. Please take them up quickly because we have lost a lot of time today.
Urvi, as far as the disposal of CSF is concerned, all the needles, all the syringes which are used, we dispose and discard them off in this so-called puncture-resistant, autoclavable discard container. So, and for each patient, we use a new sterile syringe.
Are you done answering all questions? Group 6?
Yes, sir. I believe all the questions have been addressed.
Great. Okay. Thank you so much. You can stop presenting your screen now. So I hope that